This is not. All right, fine. No, I didn't have your name over here because it's Babatunde is written by. We'll have 3 talks in this session. And the first is by Fernando da Silva Borges, who is just arrived from Barcelona, is it? Okay, so that's the title.
Thank you. My name is Fernando. I'm a postdoc at the Federal University in the Center for Mathematics, Computation, and Cognition. Today I'm going to talk about the time patterns in neural networks using the model called the adaptive exponential integrated fire model. Basically, my work, I have studied the 2 patterns, asynchronous and synchronous patterns. And this kind of the behavior is associated to epileptic seizures. There are many kinds of synchronized types, but here my type, my study is about the high level of synchronized neuronal networks.
Fernando, before we go on, you need to be tracked by this, and perhaps you need. This.
Thank you.
I'll give you 1 more minute.
Thank you very much. I will start. My name is Fernando da Silva Borges. I'm from Federal University of ABC, and I'll talk about the firing patterns in neural networks. I will talk about a specific model. It's an integrated fire model with adaptation. In my studies, I have studied synchronized patterns and asynchronized patterns. My type of synchronized type is a high synchronized. This type of synchronization is associated to epileptic seizure. For example, here I'm showing the one data from human hippocampus slices, but it's a very common pattern in rats and other animal models, and the in vivo experiment in human. There are 2 kinds of firing pattern. In the resting states, in normal states, the neurons, the principal neurons, they are most common neurons in this region, the hippocampal region, they exhibit a synchronous firing pattern. And this firing pattern in this kind of neurons is like a spike. They have a one spike. There are some time in silence, and then another few seconds, another spike. However, in this kind of synchronization, high-level synchronization, we have a seizure behavior where these principal neurons change the firing pattern from spike asynchronous to high level of burst synchronized spike. Then I'm trying here model something to understand how this could happen. But there is a question about these 2 kinds of firing patterns. Both of these firing patterns are supported for the same network. For example, in this specific case, is a hippocampal network. It's like this region is like this, and the network from this behavior don't change. It's the same network. It's the same coupling. It's the same architecture. Then, how could the same network change from this in this short time? Then, I think to model this, you have two some issues. For example, This neural network has the same structure, and they have to change from this to this, and the opposite case too, from burst to spike, without changing the structural network and the local dynamics. Only some can put some trigger to change from here to here, and the opposite case. Then what you have to do here? Find some network in the B-stable firing pattern, where asynchronous firing with spike and high synchronous firing with burst is possible. To do that, first we have some previous works, where we study how the network could change from asynchronous to high synchronous firing pattern using this integrated firing model. In both this work, we saw when we increase the excitatory probability or the synaptic conductance, excitatory synaptic conductance, we can change from asynchronous to synchronous. This is a raster plot. Each dot here represents a fire in neuron. It's a network, random network with 1,000 neurons, and it's an example of two membrane potential in two random neurons. Here it's a synchronous, it's synchronous, high level of synchronous. It's a normal, it's an epileptic pattern. Then, but in both of this work, you have increased some, we change the network. We increase the probability or change the synaptic conductance. Now, we are thinking what it's possible to change from here to here without changing the network. Then to do that, let's explain again the previous model. In this model, we use these equations, integrated fire. When the second equation here is about adaptation. It's very important here because only with this adaptation, the neuron can change from regular spike to burst. Then only increase for example, the coupling here. We have three cases, very low coupling, medium, and high coupling. And the Kuramoto parameter show us how the network is synchronized. For example, in these black dots, in the first case, is the black arrow here. We have small values of the coupling, and the network represents asynchronous behavior. When we increase the coupling, the network can show firstly spike synchronization, and when I increase more, I have burst synchronization. It's another guys told you about this parameter is very similar, and when I have average this parameter, I can talk about the general behavior of the network. If the R is equal to 1, the network is fully synchronized. What's important here how to try everything here. I was changing the network. Now I'm going to try modeling the epileptic network. To do that, I have some issues. For example, the epileptic network has to exhibit epileptic seizures at high level of synchronization. And the Traub and Wong have proposed the three more features of this network. All the neurons in the network have to able to spike in burst. There is a strong excitation in these neurons, and a relative disinhibition. It's more important for us is because from the physiological pathological networks, there are some experimental evidence showing there is a high relative disinhibition in the network, in the neural network. Then this modeling the network has support both patterns with the same structure, the same connection, the same constants show both patterns, asynchronous and synchronous. Then we have two ways to do disinhibition. Two types of the loss of inhibition. The first is a decrease of synaptic strength. And in our model, we decrease the relative inhibitory synaptic conductance, is DG. Then the inhibitory contents are decreased. And another one, if some neurons, inhibitory neurons dead, if I take off my network, these neurons, the inhibition are decreased too. Then both of these strategies, I have changed my pattern from asynchronous to high level of synchronous. And here I have a parameter space with the coupling. It's an excitatory coupling, relative inhibitory excitatory coupling. It's more important here is the white region. When I have this behavior, expected behavior, the B-stable behavior. In this network, any parameters in this white region, I have both only dependent on the initial conditions. If I put my network in the white region, I can show the asynchronous pattern and high level of the synchronous pattern. It's a kind of stereotypic behavior. If my initial condition is asynchronous, the network is still in the asynchronous behavior if the parameters are in the white region. There are two things in this B-stable region. This B-stable has a synchronous probability. Then if I put the random initial conditions in this region, I have some probability to the network synchronized. It's only near one, very near the border, the synchronized border in the yellow region. And it is robust to some noise. I can put high level in the white noise in this region. The size of the region decreases, but it's still there the B-stable region. And then if we model some aspects of this network, the first objective is to model this B-stable behavior. Now we are going to try put some aspects of the triggers. We start the network from asynchronous behavior in the B-stable regime and put some trigger. For the first trial, we put a stereo current pulse. We start randomly we started the network in this pattern. It's asynchronous, and in this asynchronous pattern, we put a square current pulse in all neurons. When we put this, we generate a asynchronous behavior. All neurons have the same input simultaneously. This stimulus have some time of duration and some amplitude. And if we are out of the B-stable region, for example, in a synchronous region, after we take off this trigger, the network stays synchronized for a few seconds, less than 0.5 seconds. It's very short time to stay synchronized. It's not a characteristic of epileptic seizure. It's only a transitory synchronization. However, in the B-stable region, where we think we model this kind of epilepsy, when we put the trigger, there are a lot of regions in the parameter space in yellow. When we put the trigger for only a few seconds, and the network stays for synchronizer maybe forever. In the yellow region, more than 10 seconds. Then here we simulate some aspects of this trigger in the epileptic neurons. We can do the opposite, stop and try to oppose the seizure, start from the synchronous, high-level synchronous behavior. We put this square current with the synchronous cohesion in initial condition. And you have now, we don't have put the serial current in all neurons, because if you put in all neurons, the synchronizer will increase and you keep the high level of synchronization. we put this current in a small fraction of neurons only. For example, fix here for a trigger for 1 second. We put this is a fraction of a neuron in the network. It has received this input. In the black region, you have the mean seizure duration less than 1 second. Then in this black region, for example, if you put 10% of neurons with this input and the amplitude equal to 20, you stop the seizure. Then you assess the epileptic seizure in the model. We try to put some negative amplitude, but in the model, the better input is positive. There are a bigger black region in this case. This paper has more things. I don't have time here, but I would like to thank my collaborators involved in this work, and another one, and this personal situation.
You didn't need the extra minute.
Yeah.
Okay. Questions? All your results are only for the model, Fernando? Or do you have any indication that this might work in practice?
Yes, this experimental evidence only supports the model, but it's a little hard to compare the model exactly. The raster plot and the firing rate, it's not so easy. But the experimental evidence is supported by qualitativity.
Okay. Murillo.
Epileptic seizure being a consequence of coexistence, the brain is coexistence is always good for a complex system, right? Because it gives you flexibility to do other things. It's so unfortunate that in this case, one of the events is of an epileptic seizure, right?
Yes.
Do you see any way of using the epileptic seizure for the own benefit of brain computing, or it's always a problem?
No, I think this case, a specific case, is a problem because we have a high level of synchronicity. In another type of the stability behavior, we have no so high synchronicity. This another kind of synchronicity is good for the brain. It's good for memory formation and information transfer. But I'm showing the bad case when the synchronized is high level. In the burst synchronized, it's the worst.
Well, thank you very much, Fernando. The next speaker is Alejandro.
Opa.
Thank you.
Linked forever. One can see that we are drawing to the end of the conference, right? Okay, so this talk is going to be on Turing pattern modulation by chemical gradients.
Okay. Thank you for the audience. First of all, sorry for my poor English, sorry. Well, I try to discuss the Turing pattern modulation. Sorry. Here. This work is developed together my students, Leonardo. We try starting now this subject to. But this work can be divided in two main points. The first point is a technical, and the second is application. In technical, for example, we use the standard Turing patterns, considering, for example, the oscillator reactions. Usually people use, for example, study the Turing pattern, consider Neumann or periodic boundary conditions, because usually people avoid the biggest strong influence of the boundary conditions in the Turing pattern. In our case, you use the Dirichlet conditions, because we need the strong influence of the boundary conditions in the Turing pattern. And the difference more is, for example, we use the small size. Usually in the literature, uses big size of the system. What is the relative big and large size? For example, you consider the average size of the Turing patterns. If the system is near this size, we name it the small relative size. But okay. We study, for example, the modulation by the Dirichlet boundary conditions. We can, for example, using the boundary conditions, can break the symmetry of the Turing pattern. I don't know if the correct symmetry breaking is a correct terms, but we invented this now, but maybe not correct. And the motivation, for example, the second point is perhaps can use this symmetry break Turing to more or less describe the initial steps of the cellular differentiation and embryogenesis. This point is inspired by this paper, this Turing's paper, the chemical base of morphogenesis. We force more or less the point, but we consider in this case the Turing pattern, the emergence of Turing pattern, like emergence of the internal structure of cells. I don't know if it's very correctly, but maybe you can consider this. In this case, we more or less, these are some more details. The modulation, we'll consider the modulation by Dirichlet boundary conditions in two systems. For example, isothermal and non-isothermal. You consider here the Brusselator reactors. We have the activator and inhibitor. In the isothermal case, because the temperature is constant. In non-isothermal version, we consider three dependent variables: the activator, inhibitor, and temperature. Okay, we can find the spatial breaking of the Turing patterns. For example, in this case, for example, in the cell division, you have a single cell in the mitosis, for example, and produce, or the embryogenesis, produce 2 cells, 4 cells, and 8 cells. More or less in this level, the cells become differentiation. And maybe the most important influence for this is the chemical environment. In this case, you consider the, okay, maybe put a gradient boundary conditions, because maybe it's most important. I don't know, in the cell differentiation process, maybe it's the chemistry is the most important things. And in this case, we put a gradient. And in particular, if the gradient contain the critical points of the general dynamics, we can induce the Turing pattern, separate two Turing patterns, different Turing patterns in the same reactor, the same system. Okay, I use the entropy production or Minkowski functional to consider to indicate the transition, but it's not important to see entropy or the other things. It's only for put idea. Okay, so I talk about this. Okay, it try to more or less display the cellular differentiation or embryogenesis. I put figures and okay. In this case, we consider the Brusselator reactors as basic reaction, but the next steps of this work, we need to substitute this reaction by other more realistic, of course. Because it's a very usual in the literature study Turing pattern by the Brusselator reaction. And because in this, only to learn about the system and we use this reaction in particular. You have here input, output. This is activator, it's the inhibitor, it's the autocatalysis. This is very important for nonlinearity of the equation. In the general, the system is more or less this. Is a square or rectangular system, maybe. It's the chemical gradient here. For example, the colors represent the chemical concentration. And okay, input, output, the reaction. The temperature is a constant. Maybe in some cases, isothermal case is a constant. In non-isothermal case, you use a dependent variable. Temperature is a dependent variable. The general equations is this, explained in detail here. For example, this is for activator and inhibitor is for temperature. Some cases I use 2 or 3 equations. This is kinetical terms. Here, more or less, is Arrhenius equations. Maybe it's no very good idea to use for variable temperature, but maybe people use this. But perhaps it's necessary to change this, but okay. The explanation, the tail, for example, this term is input, output. It's okay, it's output, okay. In the terms of reaction, this is a mass transport. This is for temperature. Then Newton's law of cooling is important for if you try to put the gradient but in temperature, but in that case I don't show here. The matter input, output input like this, the chemical enthalpy and the thermal diffusion. Very similar equations, okay? But in this case, for example, this R1, R2 depend on the temperature because the Arrhenius equations is put here. Okay, I put only this simple thing to explain why, how to solve this equation. I consider, for the epistemology 2-dimensional to describe the part in spatial time, in spatial, and in the Ruggie-Kutta for order, in the chemical reactions, nonlinear chemical reactions is the step. It's only for completeness is only the dimensional, but it's not important here. And use these parameters. Some of these parameters was collected from the literature. Maybe it's usual, maybe other no, but okay. Of course, this you consider the Brusselator, these parameters. And we find the situation, the instabilities in the spatial structure and the stability in the temporal situations, okay. In this case, we studied the stability, instability Turing. You use, for example, for for isothermal, use these parameters here, and for non-isothermal here, maybe it's similar, okay. The parameter conditions, this. It's important, for example, the initial conditions were defined randomly around the critical point. This important point here. This is a Turing pattern, consider, for example, Neumann boundary conditions. You consider this as reference, for example, this Turing pattern, there are no influence of the boundary conditions. Neumann and the periodic sinks, for example, Neumann in this case is the zero flux in the border. Usually in the literature, people use a Neumann or periodic in order to avoid any strong influence of the border. In this case, for us, is a reference only. When I put, for example, the Dirichlet boundary conditions for this specific concentration for activator and inhibitor, you have same Turing pattern, and here we have has a change of the Turing pattern. In this case, the red numbers is near to the critical point, stationary states. If you transpose this point, the Turing pattern change. This simulation is for isothermal system, it's okay for non-isothermal system. No isothermal system is three dependent variable, the activator, inhibitor, temperature. In this case, so only the activator and the inhibitor, okay? Though there are no, for example, gradient for temperature, only for concentration. In this case, don't have gradient again. In this case, for example, it's a concentration, for example, for activator 1.8, maybe not 2 points, okay. And for inhibitor 2.099, it's the border. This, the B, it's the different values, but very near. And for the C, for D, you have a strong modification. For example, we calculate the entropy total rate for A, B, C, D, E, F. You can see the very strong modification in the entropy rate in this step, for example, here. And maybe the entropy rates are very good to analyze the different pattern. Okay. These the rate entropy production. I consider these formulas, but maybe it's possible use the Minkowski functional. The other things try to compare the formulas of the Turing pattern, but this is enough. In this case, it's okay. The same system, but we put a linear gradient here. And in the same system, you have two Turing patterns for isothermal and non-isothermal. Okay? The gradient is more or less this. For this system, 0 to 30, you use this gradient, these values of U and V. For bigger system, this 0 to 60, next case, I use this gradient, the same inclination of the gradient for two cases. Okay, it's for the small and the more larger. It's artificial things we construct here. Because this implication, for example, I have a small system, in this case is a large system, is implied in the growth of the cells. The cell is increasing, in this case, it's artificial increase, of course, and it's possible to maintain the symmetry breaking of the Turing pattern, in this case. And in the other, we simply cut, for example, you have the same gradient here, but the system was defined to 0 to 30, and the cut is exactly in the point of in this value. In the same point, you cut it here, and you made the calculation for this subsystem and the other subsystem. Okay, you divide this. I put together, for example, in this case is the Turing pattern for Neumann boundary conditions. This case for Dirichlet boundary conditions, different Dirichlet boundary conditions. This case is a gradient boundary conditions. Use Dirichlet, of course. And for example, you can see in this. For example, these are cells in the, for example, cell differentiation in the laboratory, in vitro. We can consider this. There are no chemical environment. To transfer for here, you have the same Turing pattern, but with the chemical environment, different chemical environment. For example, we can consider, for example, the cell differentiation for cardiac, for epithelial, maybe depend on the chemical environment. In this case, you use the same cells, for example, the same Turing pattern, the same system, and put the gradient. It's possible with the gradient, break the symmetry of the Turing pattern. In this case, we artificially increase the problem. In this, you cut, for example, this division of the cells. It's for isothermal things. For non-isothermals, it's similar, the Neumann, Dirichlet gradient for big system, and the division. And we can consider, of course, maybe it's very strong for adapt this situation for embryogenesis of cell differentiation. We started the collaboration with experimental using this idea for the photochemistry reactions. It's more concrete thing. It's possible to obtain the Turing pattern experimentally, and we put the gradient in the light, okay, and the gradient in the light, and to study this maybe symmetry break of Turing patterns, okay? But maybe the idea of the, inspired by the Turing's paper, consider the cell differentiation or embryogenesis, you consider this more or less, for example, this is a modulation. This case you'll have, for example, the above a Neumann. You put, for example, Dirichlet for constant. You have a modulation. You can consider for different chemical environments. You have different Turing pattern, and you need to grow the division, and you need to freezing, because if you eliminate the gradient, the Turing pattern disappear. But maybe the nature, for example, the freezing process, maybe it's polymerization of protein, the other things maybe. In the other, for example, embryogenesis, maybe it's important to use the symmetry breaking, because you have need to different cells, okay? And the biogenesis, everything, no? Maybe it's you put the thermal gradient and the symmetry breaking, modulation, everything. Okay, it's very complicated thing, but okay. And final remarks, I can consider, for example, we more or less can induce the special symmetry breaking if the gradient contain critical points of the general system. Maybe, I don't know, it's sufficient, maybe it's strong here, but we need to study for more equations, for more reactions, but maybe perhaps it's possible because near to critical points, the system is near linear, and maybe it's general, I don't know, but you need to study this. This work was motivated by Turing legacy about this, maybe to describe cellular differentiation, and you need a special symmetry breaking, is increasing, division, and of course the freezing, but it's very difficult to build a dynamic for everything, for folding the protein, for the increase. Maybe it's not difficult because the system don't conserve the matter, but maybe it's not easy. But in the next step, we pretend to replace the Brusselator chemical reactions to substitute by a more realistic system. Okay, and I would like to agree. UFABC, CAPES, you need to put the number. No, forget this. There's the reference, and thank you for your patience. Thank you.
Time for a question or two. Nivedita? No, since you were asking others, I thought we'd turn the tables. No, I had one actually. You've done the modeling via, partial differential equations with Dirichlet and von Neumann. But many of these reactions, especially in a small enclosure and in biological systems, will be stochastic. How do you take noise into your picture?
Okay, it's important. Our last work is we work in the stochastic things. Is very, very important, the statistical fluctuation. For example, we study the breaking of the chiral symmetry in the systems. And yes, we need to put the stochastic things. Perhaps it's different performed because you need to build, for example, a matrix, and they consider the neighborhood, the reactions, put the diffusion, and maybe it's better than this system to consider the diffusion, because you have a big problems in this case. For example, you use the diffusion, the constant diffusion terms, but it's macroscopic terms. It's not correct. and the same kind of the Arrhenius equations. It's a problem too. Okay, thank you.
Well, thank you very much. And the final talk of this session is by Raul. Big one? Oh. Raul de Palma, Aristides. Okay. And you'll be talking on non-local coupling among oscillators mediated by traveling waves.
Yes. So, thank you all for, excuse me. My name is Raul, and first I would like to congratulate the organizers for this amazing event, and also thank you the opportunity to be presenting my work here today. So, the work that I'm going to present here today is called Coupling Among Oscillators Mediated by Fast Traveling Waves. And this work has been made in a collaboration with Professor Ricardo Viana, especially last year while I was a master's student at Federal University of Paraná. So, this is an outline of my presentation. In the first part, I'm going to give a little introduction in this subject. Then I'm going to talk about this wave-mediated coupling, our coupled oscillators model. And in the last part, I'm going to give you some numerical results on synchronization and finally my conclusions. So, what was our motivation to study this case? We all know that in nature we have many situations where systems behave like oscillators, and they often show periodic motion and periodic dynamics. And as an example, we can see the firing of neurons, the crickets chirping, and also the flashing of fireflies. So in this figure here, we can see that we can look at the mechanism of the flashing of fireflies as a clock. And in this case, each time the clock reaches the 12th position, the clock flashes, or the firefly flashes. But we also know that these kind of biological oscillators are usually found in complex populations. They are often part of populations. And as part of population, they also can interact with themselves. In that sense, I mean that the dynamic of one oscillator, the state of one oscillator can affect the state of the others. And do this, we have this nonlinear phenomena called synchronization. Excuse me. So, synchronization is a phenomenon studied since the 17th century, and first by the Dutch mathematician Christian Huygens. So, as I said, the synchronization occurs due to the interaction between the oscillators. After some time, they can adjust their rhythms and then become a synchronized system, as we can see in this representation. So this was called the sympathy of clocks, and it was observed by Huygens. So Huygens observed that he had two clocks, two pendular clocks, and in the first moment, the clocks were desynchronized like this. But due to the interaction, after some times, what he seen was this. The clocks shown a synchronized state. But we can also see this kind of behavior in more complex systems, like when we have a bunch of fireflies. In the first moment, they are all flashing in their own rhythms, we don't have any pattern here. But after some time, we can see that the fireflies became synchronized. And this situation was observed by Buck and Buck and very studied in the past years. And what happens here? So, excuse me. What happens? Each one of these fireflies flashes, and the dynamic of the other fireflies are affected by the intensity of the light that they received, of how much light they received. So after some time, they became synchronized. So this was our main motivation to study this kind of coupling, this wave-mediated coupling, because we also have the situation where crickets can synchronize their chirping. And in these states, we have a population of crickets, and they all chirp together. And in this situation, we also have the wave-mediated coupling, but in this situation, we have an acoustic wave. So, to begin with our mathematical model, we have to deal with the two kind of vectors: the positions of the oscillators, given by these vectors R, and the state variable of the system, given by this vector X. So I'm going to consider that the oscillators are embedded in this Euclidean space, so they do not move. This is our first approximation. Then I can write my equation of the time evolution of each one of the oscillators. So the evolution is given by this vector field f of x, that if we don't have the coupling, it would be just this part. But as we have the coupling, we consider also this coupling function g that depends in this scalar field that can be the pressure of acoustic wave or the intensity of electromagnetic wave. So, and also the scalar field satisfies this dampened wave equation, where this c is the propagation velocity of the wave, this zeta is a damping parameter, and in this side we have the source terms of the propagation. So, So, our next approximation will be considered that the propagation of the wave occurs in a very fast way, much more faster than the intrinsic dynamic of each one of the oscillators. And the intensity of this scalar field here becomes stationary after a very period of time. So we can discard this term right here. And the solution to the resulting equation is this one right here, where these sigma functions are green functions that are solutions to this equation right here. And solving this wave equation, we can reach these equations right here that were also calculated in this paper by Nakal in another context. So here we see that this gamma is the square root of the damping parameter. So now I have the solution to the scalar field that I am considered that couple the oscillators. Now I can put this solution right here in the equation for the time evolution of the oscillators. And as an example, I'm going to consider that we have phase oscillators, just like the Kuramoto model. And I'm also consider the Kuramoto coupling here, where I consider that the couple function G is a linear function of x. And then I can write this expression right here. And the resulting equation for the time evolution is this. It's this one. So now I have my equation, and we decided to study the synchronization in a population of oscillators of this type. So what we did is this. We decided to study a regular lattice of oscillators, and each one of these oscillators are separated by a constant distance delta. And now I have, by making a change of variables, I reach this summation right here. And now I consider that I don't sum right in the way all the oscillators. I sum first the contribution of one side of the lattice, and after that I sum the other side of the lattice. And here the natural frequencies of the oscillators are given by a Gaussian PDF, and this constant C is given by this expression that I calculated using the normalization the normalization of the green functions. So the first phenomenon, the synchronized phenomenon that I studied was the frequency synchronization. So in this plot right here, I see the observed frequencies of the oscillators and the natural frequencies. I can see that for the first curve in red, gamma is very small. And we have a synchronized state where all the oscillators have almost the same frequency. And as gamma is increased, we see that the synchronization is lost. And for a gamma 4 times bigger than the red curve, we see this unsynchronized state. Then we decide to study the phase synchronization in the lattice. So here I have the results for the mean Kuramoto order parameter as a function of gamma. And we also studied different sizes of lattice. So in the first curve, in purple, we have a lattice of only 201 oscillators. And in the yellow one curve, we have the results for the lattice of 601 oscillators. So we can see here that we can see that as gamma is increased, again, we have a loss of the synchronization. So we see a transition for a synchronized state to an unsynchronized state. And we also see that as the size of the lattice is increased, the sensibility to the variation of gamma is also increased. But why does this happen? We have to look to the form of our interaction kernel and the coupling. We have to look to the coupling. And if we remember, this constant C is given by this expression right here. So if we took the limits where eta goes to 0, and remember that gamma is the square root of eta, we reach this result right here. C is equal 1 over n minus 1, which leads us to the global coupling, this one right here. So in this situation, we have a lattice of oscillators, and each one of the oscillators are coupled with the other. So we have to take in contribution all the oscillators when we calculate the time evolution of each one of the oscillators. And In the other hand, when we take the limit to this eta being large, we have this result right here. So C is approximately 1 over 2 exponential of minus gamma L. And when we put this in our equation for the time evolution, we have this expression right here. And as we see, only the contributions of the first oscillator, the first neighbor, are taken in account. So in this case, we have, again, the lattice, but only when we take the evolution of the oscillator, we just have to consider the nearest neighbor. So we don't achieve a synchronized state because the couple is not so efficient, if you want to say. So now my conclusions. We propose a model to describe an ensemble of oscillators coupled by traveling waves, fast traveling waves. The result is a non-local coupling. Why non-local? Because we don't depend on the distance. All the oscillators are coupled. And the strength of the couple decays with the spatial distance, as we can see because of the exponential in the expression. And as a numerical application, we study the frequency and the phase synchronization in a lattice of Kuramoto-like oscillators. And we notice this transition from a synchronized state to a non-synchronized state. And this corresponds to a passage from a global type of coupling to a local coupling. So I would like to acknowledge the funding of CNPq, CAPS, and also the support from UFPR and the ICTP for this amazing event. Thank you all for your attention, and that's it.
Questions? Yes, Hilda.
I want to know. About. How is this ensemble? I mean, you never mentioned the kind of distribution of each, let's say, oscillator that you have, because, for instance, what I'm trying to say is at the end, when you come with the fact that it preserves only the nearest neighbors. What does it mean, these nearest neighbors? They are in a lattice, they are in a random network, they are, on a ring. What are they?
I didn't understand your question. Can you repeat?
How are the distribution?
What it was?
What is the geometry of your lattice? Is it a
The
square
geometry
lattice?
is a one-dimensional lattice, so it's like a wiry.
But it's a chain, it's not a ring.
No.
It's a chain. Okay.
Yes, periodic boundaries.
Then, if it's periodic boundary conditions, it's a ring.
Yeah. You can look at it as a ring.
The ring, and you're saying doesn't synchronize. The ring does synchronize at the end. The frequency synchronize.
Yes. The frequency became, yes.
Okay, is there any other question? Okay, I had a question to ask Raul. With Kuramoto, with non-local coupling, in some situations you can have these chimera states.
Yes.
Do you see any evidence of chimeras?
No, we didn't study it in our lattice. In our work, we identified it. I didn't look for it, to be honest.
Yeah. Okay.
Yet.
Okay. Well,
yes.
Yeah. It seems like an obvious thing to say. Anyway, thank you very much for the talk, and thanks to all the speakers. I have one announcement to make, and that is that the announced coffee break is from 3 to 4:30. We will abbreviate it and convene here again at 4 o'clock. So drink your coffee fast. Only one coffee. Yeah. It would be nice to see if you can find chimeras, Raul.
Yes, yes. In our group, we have already a paper on the chimeras with Professor Viana and Professor López. I think to be honest, my next
It would be nice to also take, not just the ring, the whole Turing pattern. Also,